Okay, I will start now for the DDBJ's progress. And our institute, National Institute of Genetics, is on Mishima. That is about 100 kilometers from Tokyo, on the center part of Japan. And we enjoyed beautiful Mount Fuji scenery when it's fine. And well, National Institute of Genetics have roughly two parts. One is the research departments. And we have 4 research departments. One is the Department of Informatics. I'm here, and another one, others Department of Genomics and Evolutionary Biology, and Department of Gene Function and Phenomics, and Department of Chromosome Science. And these are the research department like normal laboratories in universities. And we have another part in our institute, that is the Intellectual Infrastructure Center. And we have 3 centers in this part, and one is the Bioinformatics and the DDBJ Center. And I'm here, and I'm a head of the Division of International Affairs. And another one is the Advanced Genomics Center, that is the Sequence Center. And we have NGSs to provide the genome sequencing in Japan and other countries. And third is the Genetic Resource Center. It provides kind of the animals and plants and bacterial genome resources for the researchers around the world. And our institute is a part of the research organization of information and systems. And there is a 4 national institute in this organization. One is National Institute of Polar Research, and the National Institute of Informatics, and the Institute of Statistical Mathematics, and us, NIG. And these 4 institutes makes one research organization for the information and systems. And moreover, including these, the research organization and other national institutes forms one big university, graduate university. The Graduate University for Advanced Studies. We call it Sokendai. And 15 national institute and 1 individual faculties makes world-class university in Japan. And in our lab, we have some ongoing genome projects, and like algae and cricket and rice genomes, rice GWAS project, and cats genome project, and T3, thousands of T3s GWAS project, and wasabi, that is the Japanese horseradish. And tea and wasabi is very important for sushi. And strawberry is other big project. And in our NIG's place Shizuoka University near the Mount Fuji, and tea and wasabi and strawberries are the local specialties in Shizuoka Prefecture. And so we have the big project for the local specialties genome project. And we also provide web tools and genome databases. One is the Maple base for the liverwort genome, and another is the Felscatus genome database, CATS genome database. And we also provide DFAST, that is the very fast bacteria and virus annotation tools. And we make the COVID-19 genome annotations using the DFAST. And that is the very brief introduction of my lab. But today, I'd like to introduce DDBJ's progress. And DDBJ stands for DNA Data Bank of Japan, you know. And DDBJ is the partner of the INSDC. INSDC stands for the International Nucleotide Sequence Database Collaboration with the NCBI in US and UNA in EU. And already presented the INSDC stand very long. The INSDC is very long standing database collaboration in the world. And DDBJ release 1 was provided in 1987, and we exchanged it with the EMBO and GenBank over 30 years. And so DNA data banks was briefly submitted nucleotide, user submit the nucleotide sequence, and we check its data, metadata carefully. And if it's okay, we put it into the database, the big one, the ISDC database, and we make it public via the internet. That is the business of DNA databanks. And we have several kinds of databases, including among the ISDC. One is the long-standing annotated sequence database. You know the very famous one, GenBank, and ENA and DDBJ. And another one is the capillary, but it's getting old, and it will be vanished in the near future. And the biggest one is the NGS Reads one, SRA, you know, sequence read archives. And we also have to bind several types of data and study database and sample database, you know, bioproject database and biosample database. And another one is the assembly database. This database provides the assembly information for many assembled genomes. And we exchange completely among the INSDC in this database. But outside of INSDC, INSDC-related database exists, and functional genomes data, GEO and Express, and GA, and variation database for human, and genotype and phenotype databases for human. And outside of INSDC, we exchange the metabolomics dataset within ENA and EBI and DDBJ. Metabolites and Metabank. I will introduce after. And about 140 people work for the INSDC. It is a very big collaboration project, and so every year we have the INSDC, the technical meeting in every May in every year. But from 2020, we cannot gather because of the COVID. But we have the INSDC meeting on Zoom in 2020, and this year around 1 week, we exchanged the information and discussed how to collect and how to describe the data and metadata. And I will introduce briefly the several types of database in the INSDC. The one is the TRAD INSDC, and this part, GenBank and ENA and DDBJ. And this is an example of the TRAD database in the INSDC. And these are the metadata and locus and excision and difference. Of course, the lineage and organism and annotations, and the last part is the data itself. And the trial analysis, sorry, it is described in Japanese, but well, we have the over 15,000 billion bases, and for the files entries, we have 28,000 million sequences. So it becomes a very big database. But the NGS is more bigger than the databases. And NGS reads, we provide the SRA, sequence read archive. And this graph is always shown by such kind of talk. And you know, the cost per log megabase of DNA sequence went down dramatically around 2007. What happened in 2007 is the emerging of the next generation sequences. And the first NGS, Solexa, emerged in 2007, and now we have the PacBio, Secure2, and Illumina NovaSeq, and Oxford Nanopore, such kind of very high throughput sequencers we can use, and we have the NGS database for these data. And as of today, we have 27 database, open database data in SRA for open access data. And NCBI have the control access data in SRA, the human data, and the total they have the 60 petabytes of data in SRA. But open access data as the INSDC's open data, we have 27 petabytes, but it is very big data. And well, that is the brief introduction of the overall the INSDC. And I will describe bioinformation and DDBJ center updates in this few years. And okay, the personnel, you can see this personnel in DDBJ's website, and 43 people are worked for DDBJ at this moment. And as faculties, we have 9 personnel, and we have 3 coordinators at the head of the database division, and we have 13 curators for the curation of many types of the data. And as a system engineer, we have around 20 people in DDBJ, and first we should describe the response of to the COVID-19 in DDBJ. And from started the reaction of COVID, we have been work from home mode since the March 2020, and so the curators mainly work from home in this from the COVID occurs. And currently, the half of people coming back to the office, but the work from still the many people work from home in DDBJ. And curation and engineering and programming can done from home, and so the DDBJ is still from work from home mode. And we are in collaboration with the COVID-19 data portal Japan with the National Institute of Informatics and other institute, and we collect and curate and provide the COVID-19 and the data and metadata as the COVID-19 data portal Japan. And also we started the whole sequencing for the SARS-CoV-2 genome sequence with our local government, Shizuoka Prefecture, and we make a memorandum of cooperation with Shizuoka Prefecture for the implementation of whole genome analysis of novel coronavirus from this April, and we started the SARS-CoV-2 genome sequence sequence and correction, and provide to the INSDC and GISAID. We already submit to INSDC and GISAID over the several hundreds of the whole genome sequence of SARS-CoV-2 from this local government sample. And we started to provide new DBs and services in these few years. And we started functional genomics database and variation database and metabolomics database. And first is the new database, Genomic Expression Archive, GEA. That is the long-waiting one, and that is we have launched a long-awaiting database. And for the functional genomics data, that is the counterpart of GEO and AliExpress. And this archives the data in the standard image tab format and metadata, and that is indexed by EBIomics DI, and so you can search our metadata from the EBIomics DI, and when you find our GES data, and you can get omics data from us in GA database. And another one is Japan Variation Archive, JVAR, and we have launched two new database, Japan Variation Archive JVAR for human variation data. The one is the JVAR SNP, that is counterpart of DBSNP. And another one is JVAR SB, that provide structural variation for human. That is the counterpart of DBBAR. And JVAR accept human data only as like NCBI. The EBI's variation database, EVA and DGVA accept non-human data, but we only accept human data only. That is the limitation of our personnel and our performance. And another one, the last very the new database is from in October 2020. We have released Metabobank for the metabolomics data as a member of Metabolomics Exchange, and we currently accept from the metabolomics data from RIKEN and Kazusa DNA Research Institute, and we will release soon. We started to release soon metabolomics data and exchanging data as a member of the Metabolome Exchange. And so we construct the first DDBJ first annotation submission tool made by Tanizo Yasuhiro. He is our lab's assistant professor, and he started this service for his own interest. But this pipeline is very useful, and it becomes now official submission tool in DDBJ. And currently we have the quality control function in DDBJ, and taxonomic assignment of genome is checked by using average nucleotide identity, ANI. And in the last year, 92% of bacterial whole genomes were annotated and submitted through DFAST in DDBJ. And so it becomes our DDBJ's very good tools for submission, and we are now expanding the first to the RG or more eukaryote annotation will be annotated using the DFAST. And using the DFAST, we accept the 10K microbe type strain project, and we already released genome sequence data of more than currently we accept more than 1,000 type strain microbe type strain data from IMCAS as the effort of WDCM sequence project, and we commit deeply for the 10K microbe type strain project, and help to provide the data for the ISDC and for the world researchers for the microbes. And it is the small changes, but website are renewal in 2021 and in DDBJ, and we have revised our terms of use at February in this year in response to the EU GDPR. And for the educational activity, education and lecture activity, we have the 15 activities in 2018 and 2019 for each year, but 2020, it is numbers decreased for the COVID-19, but we mainly we provide several educational and lecture activity online using Zoom and other e-learning system, and we still provide learning activities for how to use the INSDC data. And finally, energy supercomputer updates. And we replaced our supercomputer in 2019. And now we have over 11,000 cores and 43.6 petabyte HSM storage. And the very good features, we provide a Singularity and Docker container images on our supercomputers. And as you may know, the container system is now a very useful equipment for the even the small computer and supercomputers. And general VM, old traditional VM is like ESXi. All whole systems launch on the Linux system, but like a Docker and Singularity container, the kernel are shared in the normal system and the container systems. And Docker is very famous container system, but Docker needs the root super user support. But Singularity, if you use Singularity, normal user can launch the container system. And moreover, the Singularity can use the Docker container. And so we provide Singularity container system in our supercomputer, and you can use over 2,000 Docker container images at biocontainers. And we provide all of biocontainers container in our supercomputer. And so it is very useful container system on our supercomputer. And also we started interconnection with public cloud and AWS. And we connect the SINET using the SINET 5 to AWS from our supercomputer. And inbound and outbound speed is 10 Gbps, and so that means you can transform the 100 tera per day. That is very fast and very good connection. And the AWS network fee and 75% off, and so it is a very good bargain price for the connect to the AWS. And moreover, we have the scientific network interconnection of the several national supercomputer in Japan, and so we have provided a scientific big data repository and high throughput data access, and also we provide VM and container system using this interconnect. And also we have a secure partition for the personal genome analysis. And it is the highly secure part for the human genome analysis. And we provide databases and tools and supercomputers, and that is a unique analysis platform for researchers in Japan and world. And finally, we also provide semantic representation of DDBJ, RDF style of DDBJ are released, and so you can easily connect the DDBJ data for like Interpol and other RDF resources easily. And so, we help to reuse the scientific data using such kind of the databases, tools, and supercomputer. And 감사합니다 and share for your listening. Thank you.
Thank you.
Thank you for a nice talk. May I ask you a quick question? May I ask your sharing or data deposit policy in Japan?
Excuse me?
I'm curious about data sharing or data deposit policy in Japan. So how researchers are depositing in Japan?
Well, I feel you and Korean and Chinese data opening policy is very solid and very good. But in Japan, basically, things are getting better and better, but still researchers tend to close their data, but for the sequence data, you should submit to DDBJ when you make a publication, and it's a good. And but other other data, we still need more. We should push we should encourage to open data. It is our situation.
Thank you.
Any other questions or comments for Dr. Nakamura? 1, 2. Okay, if not, thank you, Dr. Nakamura, and that will end session 1.